So today discuss we about the cancer. So there are certain genes that are responsible in formation of the cancer. So so four main classes of genes are altered frequently in cancer. So these are four main classes of genes that are frequently altered in cancer. So suppose the proto-oncogene, the mutate form of proto-oncogene is called it as oncogene, and it loses its normal function and leads to cancer. Loss of the functional loss of tumor suppressor gene leads to formation of the cancer. mRNA genes, there are certain roles of the mRNA genes. When the microRNA or mRNA is mutated in such a way that it loses its normal function and leads to a cancerous cells. There is some mutator genes. So these mutator genes have some important functions in the regulations of cell division and growth. But when those mutator genes are mutated in such a way that it also loses its normal functions, and the cell become cancerous. So first we discuss on proto-oncogene. So name is proto-oncogene stimulate cell proliferations. So there are certain genes in our body, those are called proto-oncogene, and the product of the proto-oncogene encodes some product that helps in stimulations of the cell proliferation, cell division and growth, normal growth of the organism. But when a proto-oncogene is mutated in some way, it called it oncogene, and when the proto-oncogene is transformed into oncogene, it loses in normal function. So there are oncogenes. So there are two roles of the oncogene. The product which is formed from the oncogenes are much more than the product of normal proto-oncogene. So active more than normal. So proto-oncogene is active only in certain cases, or if it is activated, it is regulated in a steady way. It cannot produce a large amount of the product. So when proto-oncogene is transformed in oncogene, it loses its normal functions and it activates more than the normal proto-oncogene and activate at inappropriate time. So when proto-oncogene is transformed into oncogenes, it active more than the normal proto-oncogene and activate at inappropriate time. So there is no particular time in which the oncogene is activated. Maybe oncogene is activated all the way constitutively expressed. So now the next tumor suppressor gene. So normally the function of the tumor suppressor gene is to inhibit cell proliferation. So if we have to inhibit the cell proliferations, we use a tumor suppressor gene. So main role tumors, so when a cell became proliferations abnormally, in this case tumor suppressor gene encodes some protein or activates some transcription factor like p53 p21 that inhibit the cell proliferations. But when the tumor suppressor gene is mutated, so when the tumor suppressor gene mutated, it loses its normal functions and so lost their inhibitory functions and cell become cancerous. So mutant form of tumor suppressor gene cannot work properly and it cannot able to inhibit their inhibitory function. Now mRNA gene. So we find the mRNA genes in the RNA interference, mRNA and siRNA. So when certain viral or foreign DNA RNA enter in our body evolves some defense mechanism by which it inhibits the transcriptions of those DNA or silenced the synthesis of those mRNA. So the normal mRNA genes in our body inhibit translations inhibit translation of mRNA encoded by other genes, not our body, the mRNA which is encoded by other genes, and the role of the miRNA is to inhibit their translations process or maybe transcription. Now the next is now when the miRNA is overexpressed, so when the miRNA is overexpressed, so overexpressed miRNA genes act as oncogene. Overexpressed, so when suppose the miRNA which is present in our body cell is transformed in such a way that it leads it expressed constitutively or it is overexpressed, so it acts like a oncogene. But when the mRNA genes are underexpressed, it acts like defective tumor suppressor gene. So the miRNA gene, the normal function of the mRNA gene is to inhibit the translations or transcriptions translations of the mRNA which is encoded by other genes, or it may leads to silence of the foreign DNA or foreign genes. But when overexpressed mRNA acts as an oncogene and underexpressed defective tumor suppressor gene. Now next important gene, that is mutator gene. So the product of wild type mutator genes have two functions: fidelity of replications and maintenance of genome integrity. So fidelity of replications means you can say that error-free replication or maybe less error replication. But when the replications occurs, the DNA polymerase which synthesizes the daughter strand that leads some error during the replication process, which is proofreaded by the DNA polymerase itself. But in this proofreading activity, or there are several type of repair mechanisms or protein that helps in the repair of that error replications. So mutator gene is such a gene that helps in error free, not actually error free, less error replications, or you can say the fidelity of replication, that whenever the error occur, the mutator gene helps in the repair process. Maintenance of genome integrity, because you have to maintain the genome integrity. If genome integrity does not maintain, the gene sequences or the genome is changed from generation to generation. So it helps in maintenance of genome integrity. Fidelity of replication and maintenance of genome integrity is depend on each other, because if the fidelity of replication does not maintain, the maintenance of genome integrity is impossible. So mutator gene helps in those process. So fidelity of replication is maintained by mutator gene, and also maintenance of genome integrity is also maintained by mutator gene. That are the two processes. So now when the mutator gene is mutated, so mutator genes, cells accumulate mutational error in any gene including proto-oncogene and tumor suppressor gene. Cells accumulate mutational error. What is mutational error? Mutational error in any gene, including it, the mutational errors may occur in the proto-oncogene or in tumor suppressor gene. If mutational error occurs in the proto-oncogene may transform into a oncogene. And if the mutational error occurs in tumor suppressor gene, the tumor suppressor loses inhibitory function and ultimately cell becomes the cancerous. So cells accumulate mutation because the mutational error cannot repaired, because there is a mutation in mutated gene. So mutator genes unable to repair the mutational error, and that leads to accumulates the mutational error in any types of gene in our body, including proto-oncogene or tumor suppressor gene. Thank you. If you like the video, please share with your friends.